Algae.
Algae are a very large and diverse group of eukaryotic organisms, ranging from unicellular genera such as Chlorella and the diatoms to multicellular forms such as the giant kelp, a large brown alga that may grow up to 50 meters in length. Most are autotrophic and lack many of the distinct cell and tissue types found in land plants such as stomata, xylem and phloem. The largest and most complex marine algae are called seaweeds, while the most complex freshwater forms are the Charophyta, a division of algae that includes Spirogyra and the stoneworts. There is no generally accepted definition of algae. One definition is that algae have chlorophyll as their primary photosynthetic pigment and lack a sterile covering of cells around their reproductive cells. Other authors exclude all prokaryotes and thus do not consider cyanobacteria, blue-green algae, as algae. Algae constitute a polyphyletic group since they do not include a common ancestor, and although their plastids seem to have a single origin, from cyanobacteria, they were acquired in different ways. Green algae are examples of algae that have primary chloroplasts derived from endosymbiotic cyanobacteria. Diatoms are examples of algae with secondary chloroplasts derived from an endosymbiotic red alga. Algae exhibit a wide range of reproductive strategies, from simple asexual cell division to complex forms of sexual reproduction. Algae lack the various structures that characterize land plants, such as the folids, leaf-like structures, of bryophytes, rhizoids in non-vascular plants, and the roots, leaves, and other organs that are found in tracheophytes, vascular plants. Most are phototrophic, although some groups contain members that are mixotrophic, deriving energy both from photosynthesis and uptake of organic carbon either by osmotrophy, mesotrophy, or phygotrophy. Some unicellular species such as the green algae Prototheca and Helicosporidium, and many euglenids and dinoflagellates, have become heterotrophs, sometimes parasitic, relying entirely on external energy sources and have limited or no photosynthetic apparatus. Some other heterotrophic organisms, like the apicomplexans, are also derived from cells whose ancestors possessed plastids, but are not traditionally considered as algae. Algae have photosynthetic machinery ultimately derived from cyanobacteria that produce oxygen as a byproduct of photosynthesis, unlike other photosynthetic bacteria such as purple and green sulfur bacteria. Fossilized filamentous algae from the Vindor Basin have been dated back to 1.6 to 1.7 billion years ago.
Etymology and study.
The singular alga is the Latin word for a particular seaweed and retains that meaning in English. The etymology is obscure. Although some speculate that it is related to Latin alga, be cold, there is no known reason to associate seaweed with temperature. A more likely source is alga, binding, entwining. The ancient Greek word for seaweed was phukos, or phycos, which could mean either the seaweed, probably red algae, or a red dye derived from it. The Latinization, fucus, meant primarily the cosmetic rouge. The etymology is uncertain, but a strong candidate has long been some word related to the biblical pvk, puke, paint, if not that word itself, a cosmetic eyeshadow used by the ancient Egyptians and other inhabitants of the eastern Mediterranean. It could be any color, black, red, green, blue. Accordingly, the modern study of marine and freshwater algae is called either phycology or algology, depending on whether the Greek or Latin root is used. The name fucus appears in a number of taxa.
Classification.
Most algae contain chloroplasts that are similar in structure to cyanobacteria. Chloroplasts contain circular DNA like that in cyanobacteria and presumably represent reduced endosymbiotic cyanobacteria. However, the exact origin of the chloroplasts is different among separate lineages of algae, reflecting their acquisition during different endosymbiotic events. The table below describes the composition of the three major groups of algae. Their lineage relationships are shown in the figure in the upper right. Many of these groups contain some members that are no longer photosynthetic. Some retain plastids, but not chloroplasts, while others have lost plastids entirely. Phylogeny based on plastid not nucleocytoplasmic genealogy. Linnaeus, in Species Plentrum, 1753, the starting point for modern botanical nomenclature, recognized 14 genera of algae, of which only 4 are currently considered among algae. W. H. Harvey, 1811, 1866, and Lamoureux, 1813, were the first to divide macroscopic algae into 4 divisions based on their pigmentation. This is the first use of a biochemical criterion in plant systematics. Harvey's 4 divisions are: red algae, Rhodophyta; brown algae, Heteromonophyta; green algae, Chlorophyta; and Diatomaceae. At this time, microscopic algae were discovered and reported by a different group of workers, for example, O. F. Muller and Ehrenberg, studying the infusoria, microscopic organisms. Unlike macroalgae, which were clearly viewed as plants, microalgae were frequently considered animals because they are often motile.
Relationship to higher plants.
The first plants on Earth probably evolved from shallow freshwater charophyte algae much like Charophyta almost 500 million years ago. These probably had an isomorphic alternation of generations and were probably filamentous. Fossils of isolated land plant spores suggest land plants may have been around as long as 475 million years ago.
Morphology.
A range of algal morphologies are exhibited, and convergence of features in unrelated groups is common. The only groups to exhibit three-dimensional multicellular thalli are the reds and browns, and some chlorophytes. Apical growth is constrained to subsets of these groups, the floridiophyte reds, various browns, and the charophytes. The form of charophytes is quite different to those of reds and browns, because they have distinct nodes, separated by internode stems. Whorls of branches reminiscent of the horsetails occur at the nodes. Conceptacles are another polyphyletic trait. They appear in the coralline algae and the hildebrandiales, as well as the browns. Most of the simpler algae are unicellular flagellates or amoeboids, but colonial and non-motile forms have developed independently among several of the groups. Some of the more common organizational levels, more than one of which may occur in the life cycle of a species, are colonial, small, regular groups of motile cells; capsoid, individual non-motile cells embedded in mucilage; cocoid, individual non-motile cells with cell walls; palmaloid, non-motile cells embedded in mucilage; filamentous, a string of non-motile cells connected together, sometimes branching; parenchymatous, cells forming a thallus with partial differentiation of tissues. In 3 lines even higher levels of organization have been reached, with full tissue differentiation. These are the brown algae, some of which may reach 50 meters in length, kelps, the red algae, and the green algae. The most complex forms are found among the green algae, sea chorales and Charophyta, in a lineage that eventually led to the higher land plants. The point where these non-algal plants begin and algae stop is usually taken to be the presence of reproductive organs with protective cell layers, a characteristic not found in the other alga groups.
Physiology.
Many algae, particularly members of the Charophyta, have served as model experimental organisms to understand the mechanisms of the water permeability of membranes, osmoregulation, turgor regulation, salt tolerance, cytoplasmic streaming, and the generation of action potentials. Phytohormones are found not only in higher plants, but in algae too.
Symbiotic algae.
Some species of algae form symbiotic relationships with other organisms. In these symbioses, the algae supply photosyntates, organic substances, to the host organism providing protection to the algal cells. The host organism derives some or all of its energy requirements from the algae. Examples are as follows.
Lichens.
Lichens are defined by the International Association for Lichenology to be an association of a fungus and a photosynthetic symbiont resulting in a stable vegetative body having a specific structure. The fungi, or mycobionts, are mainly from the Ascomycota with a few from the Basidiomycota. They are not found alone in nature, but when they began to associate is not known. One mycobionte associates with the same phycobionte species, rarely two, from the green algae, except that alternatively the mycobionte may associate with a species of cyanobacteria, hence photobionte is the more accurate term. A photobionte may be associated with many different mycobiontes or may live independently. Accordingly, lichens are named and classified as fungal species. The association is termed a morphogenesis because the lichen has a form and capabilities not possessed by the symbiont species alone. They can be experimentally isolated. It is possible that the photobionte triggers otherwise latent genes in the mycobionte.
Coral reefs.
Coral reefs are accumulated from the calcareous exoskeletons of marine invertebrates of the order Sciractinia, stony corals. As animals they metabolize sugar and oxygen to obtain energy for their cell-building processes, including secretion of the exoskeleton, with water and carbon dioxide as byproducts. As the reef is the result of a favorable equilibrium between construction by the corals and destruction by marine erosion, the rate at which metabolism can proceed determines the growth or deterioration of the reef. Dinoflagellates, algal protists, are often endosymbionts in the cells of marine invertebrates, where they accelerate host cell metabolism by generating immediately available sugar and oxygen through photosynthesis using incident light and the carbon dioxide produced by the host. Stony corals that are reef-building corals, hermatypic corals, require endosymbiotic algae from the genus Symbiodinium to be in a healthy condition. The loss of Symbiodinium from the host is known as coral bleaching, a condition which leads to the deterioration of a reef.
Sea sponges.
Green algae live close to the surface of some sponges, for example, breadcrumb sponge, Halichondria peniса. The alga is thus protected from predators. The sponge is provided with oxygen and sugars which can account for 50 to 80% of sponge growth in some species.
Life cycle.
Rhodophyta, Chlorophyta and Heterocontyphyta, the three main algal phyla, have life cycles which show tremendous variation with considerable complexity. In general there is an asexual phase where the seaweed cells are diploid, a sexual phase where the cells are haploid followed by fusion of the male and female gametes. Asexual reproduction is advantageous in that it permits efficient population increases, but less variation is possible. Sexual reproduction allows more variation, but is more costly. Often there is no strict alternation between the sporophyte and also because there is often an asexual phase, which could include the fragmentation of the thallus.
Numbers.
The algal collection of the U.S. National Herbarium, located in the National Museum of Natural History, consists of approximately 320,500 dried specimens, which, although not exhaustive, no exhaustive collection exists, gives an idea of the order of magnitude of the number of algal species. That number remains unknown. Estimates vary widely. For example, according to one standard textbook, in the British Isles the UK Biodiversity Steering Group report estimated there to be 20,000 algal species in the UK. Another checklist reports only about 5,000 species. Regarding the difference of about 15,000 species, the text concludes, it will require many detailed field surveys before it is possible to provide a reliable estimate of the total number of species. Regional and group estimates have been made as well. 5,000 to 5,500 species of red algae worldwide, some 1,300 in Australian seas, 400 seaweed species for the western coastline of South Africa, and 212 species from the coast of KwaZulu-Natal. Some of these are duplicates as the range extends across both coasts, and the total recorded is probably about 500 species. Most of these are listed in List of Seaweeds of South Africa. These exclude phytoplankton and crustose corallines, 669 marine species from California, U.S., 642 in the checklist of Britain and Ireland, and so on, but lacking any scientific basis or reliable sources. These numbers have no more credibility than the British ones mentioned above. Most estimates also omit microscopic algae, such as phytoplankton. The most recent estimate suggests a total number of 72,500 algal species worldwide.
Distribution.
The distribution of algal species has been fairly well studied since the founding of phytogeography in the mid-19th century AD. Algae spread mainly by the dispersal of spores analogously to the dispersal of plantae by seeds and spores. Spores are everywhere in all parts of the earth, the waters fresh and marine, the atmosphere, free-floating and in precipitation or mixed with dust, the humus and in other organisms, such as humans. Whether a spore is to grow into an organism depends on the combination of the species and the environmental conditions of where the spore lands. The spores of freshwater algae are dispersed mainly by running water and wind, as well as by living carriers. The bodies of water into which they are transported are chemically selective. Marine spores are spread by currents. Ocean water is temperature selective, resulting in phytogeographic zones, regions and provinces. To some degree the distribution of algae is subject to floristic discontinuities caused by geographical features, such as Antarctica, long distances of ocean or general land masses. It is therefore possible to identify species occurring by locality, such as Pacific algae, or North Sea algae. When they occur out of their localities, it is usually possible to hypothesize a transport mechanism, such as the hulls of ships. For example, Ulva reticulata and Ulva fasciata traveled from the mainland to Hawaii in this manner. Mapping is possible for select species only. There are many valid examples of confined distribution patterns. For example, Chlorella is an Arctic genus and is not mapped far south of there. On the other hand, scientists regard the overall data as insufficient due to the difficulties of undertaking such studies.
Locations.
Algae are prominent in bodies of water, common in terrestrial environments and are found in unusual environments, such as on snow and on ice. Seaweeds grow mostly in shallow marine waters, under 100 meters, 330 feet. However, some have been recorded to a depth of 360 meters, 1,180 feet. The various sorts of algae play significant roles in aquatic ecology. Microscopic forms that live suspended in the water column, phytoplankton, provide the food base for most marine food chains. In very high densities, algal blooms, these algae may discolor the water and outcompete, poison, or asphyxiate other life forms. Algae are variously sensitive to different factors, which has made them useful as biological indicators in the Ballantine scale and its modification.
Cultural associations.
In classical Chinese, the word zhao is used both for algae, and, in the modest tradition of the imperial scholars, for literary talent. The third island in Kunming Lake beside the Summer Palace in Beijing is known as the Zhejiang Tangdao, which thus simultaneously means Island of the Algae Viewing Hall, and Island of the Hall for reflecting on literary talent.
Uses. Agar.
Agar, a gelatinous substance derived from red algae, has a number of commercial uses. It is a good medium on which to grow bacteria and fungi as most microorganisms cannot digest agar.
Alganates.
Between 100,000 and 170,000 wet tons of macrocystis are harvested annually in New Mexico for alginate extraction and abalone feed.
Energy source.
To be competitive and independent from fluctuating support from local policy on the long run, biofuels should equal or beat the cost level of fossil fuels. Here, algae-based fuels hold great promise, directly related to the potential to produce more biomass per unit area in a year than any other form of biomass. The break-even point for algae-based biofuels is estimated to occur by 2025.
Fertilizer.
For centuries seaweed has been used as a fertilizer. George Owen of Heenley's writing in the 16th century referring to driftweed in South Wales. Today, algae are used by humans in many ways. For example, as fertilizers, soil conditioners and livestock feed. Aquatic and microscopic species are cultured in clear tanks or ponds and are either harvested or used to treat effluents pumped through the ponds. Alga culture on a large scale is an important type of aquaculture in some places. Meal is commonly used as a soil conditioner.
Nutrition.
Naturally growing seaweeds are an important source of food, especially in Asia. They provide many vitamins including A, B1, B2, B6, niacin and C, and are rich in iodine, potassium, iron, magnesium and calcium. In addition, commercially cultivated microalgae, including both algae and cyanobacteria, are marketed as nutritional supplements, such as spirulina, chlorella and the vitamin C supplement, unicellular, high in beta-carotene. Algae are national foods of many nations. China consumes more than 70 species, including fat choy, a cyanobacterium considered a vegetable. Japan, over 20 species. Ireland, dulse. Chile, cochayuyo. Lava is used to make lava bread in Wales where it is known as baralawa. In Korea, jim. In Japan, nori and anori. It is also used along the west coast of North America from California to British Columbia, in Hawaii and by the Maori of New Zealand. Sea lettuce and badlocks are a salad ingredient in Scotland, Ireland, Greenland and Iceland. The oils from some algae have high levels of unsaturated fatty acids. For example, Pyritrochloris incisa is very high in arachidonic acid, where it reaches up to 47% of the triglyceride pool. Some varieties of algae favored by vegetarianism and veganism contain the long-chain, essential omega-3 fatty acids, docosahexaenoic acid, DHA, and eicosapentaenoic acid, EPA. Fish oil contains the omega-3 fatty acids, but the original source is algae, microalgae in particular, which are eaten by marine life such as copepods and are passed up the food chain. Algae has emerged in recent years as a popular source of omega-3 fatty acids for vegetarians who cannot get long-chain EPA and DHA from other vegetarian sources such as flaxseed oil, which only contains the short-chain alpha-linolenic acid, ALA.
Pollution control.
Sewage can be treated with algae, reducing the usage of large amounts of toxic chemicals that would otherwise be needed. Algae can be used to capture fertilizers and runoff from farms. When subsequently harvested, the enriched algae itself can be used as fertilizer. Aquariums and ponds can be filtered using algae, which absorb nutrients from the water in a device called an algae scrubber, also known as an ATS. Agricultural Research Service scientists found that 60 to 90% of nitrogen runoff and 70 to 100% of phosphorus runoff can be captured from manure effluents using a horizontal algae scrubber, also called an algal turf scrubber, ATS. Scientists developed the ATS, which are shallow, 100-foot raceways of nylon netting where algae colonies can form, and studied its efficacy for 3 years. They found that algae can readily be used to reduce the nutrient runoff from agricultural fields and increase the quality of water flowing into rivers, streams, and oceans. The enriched algae itself also can be used as a fertilizer. Researchers collected and dried the nutrient-rich algae from the ATS and studied its potential as an organic fertilizer. They found that cucumber and corn seedlings grew just as well using ATS organic fertilizer as they did with commercial fertilizers. Algae scrubbers, using bubbling upflow or vertical waterfall versions, are now also being used to filter aquariums and ponds.
Bioremediation.
The algae Stichococcus bacillaris has been seen to colonize silicone resins used at archaeological sites, biodeteriorating the synthetic substance.
Pigments.
The natural pigments produced by algae can be used as an alternative to chemical dyes and coloring agents.
Stabilizing substances.
Carrageenan, from the red alga Chondrus crispus, is used as a stabilizer in milk products.